Welcome to the webinar brought to you by the IQ Patient-centric Sampling Working Group. It's my honor to be the moderator of today's webinar. My name is Zhang. I'm currently the Senior Director in the Department of Global PK/PD and Pharmacometrics at Eli Lilly, and also the co-chair of this IQ Patient-centric Sampling Working Group. Now it's my pleasure to introduce our speakers for today's webinar. Our first speaker is Parag Kumar. Parag is currently a senior clinical pharmacologist at Gilead Sciences, where he develops virology compounds. Prior to his current role, Parag served as the associate director in the clinical pharmacology at Otsuka Pharmaceutical and director of the clinical pharmacokinetic research unit of the NIH, supporting clinical pharmacology studies across the 27 research institutes of the NIH. Parag received his doctorate in pharmacy from the University of Georgia and also completed a 2-year clinical research and drug development fellowship at the University of North Carolina and Research Triangle Park in North Carolina. Parag has been a very active and valuable member of this IQ Patient-centric Sampling Working Group. Our second speaker is Melanie Anderson. Melanie is a principal scientist at Merck with over 20 years experience in clinical development. In her current role, she evaluates and implements patient-centric sampling approaches for drug-level quantitation and biomarker testing in clinical research. She recently co-led a cross-functional team at Merck working to operationalize decentralized clinical trial technology. Melanie received her bachelor degree in chemistry from Hastings College and her master in chemistry from Lehigh University. Melanie is the co-chair of the IQ Patient-centric Sampling Group. I have the pleasure to work very closely with her, and our working group wouldn't have been successful without her strong leadership. She currently also co-chair the PCSIG working group on diagnostics. Without further ado, I'll give the floor to our speakers. Parag.
Thank you, Cindy, and welcome everyone to our webinar entitled Leveraging Patient-Centric Sampling for Clinical Drug Development in Decentralized Clinical Trials, Promise to Reality. So the outline of the webinar, I'll start off. I'll go over some of the value proposition as well as introducing some of the devices that are available and considerations for selecting a device for patient-centric sampling. After that, Melanie will go into real-world case examples from various companies that have implemented this into their drug development programs, and then we'll end with a Q&A session. So let's get started. In terms of what we think about when we think about what is patient-centric sampling, we first have to define that by contrasting that to what we consider standard or traditional venous blood sampling. So this is often painful, would almost always require a visit to a clinical site. It is burdensome to participants and caregivers. It will require someone to who's trained in collection of the blood, and there are a number of restrictions on cold chain storage and shipping related to traditional blood sampling. And so in contrast, patient-centric sampling is really any sort of sampling that's less invasive than a typical blood draw. Usually it's far less painful. Often requires a lower blood volume, or what we call microsampling. And one of the key sort of benefits we have is that you can actually often collect the sample either in the patient's home or somewhere local to them. And that can either mean the patient could collect the sample by themselves, or a caregiver could assist them, or potentially something like an at-home nurse could come and collect the sample for them. So when we talk about patient-centric sampling, it's not really a necessarily new concept. It's been around since at least the 1960s. Here we have this publication by Dr. Guthrie in 1960 using dry blood sampling for heel pricks in neonates for measuring phenylalanine. And so this concept has been around for a while. Microsamples have been used in the industry previously very abundantly in the preclinical space, and that was primarily to limit animal sacrifice and enable serial sampling from single animals. However, there have been a number of challenges, especially when it comes to ensuring consistent blood volumes and accounting for a variable hematocrit that's limited our use in the clinic. However, recently there are a number of new devices, and I'll go over some of these in a few moments, but these devices have addressed many of these samples and many of these issues, and that includes standardizing the sample volume and ensuring the sample quality. And then lastly, since the COVID pandemic, we have seen a really huge enhancement in the interest in doing some sort of remote sampling or reducing the amount of time that participants are confined to the clinic for clinical trials. So in terms of the devices, as I said, there's a number of new devices. So here on the left is kind of what we think of traditional dry blood spotting on a card. And as I mentioned, there's a number of issues related to hematocrit variability that you have to account for that can reduce the accuracy in the analysis. And since then, there's been some newer technologies. One is called VAMS, volumetric absorptive microsampling. And so with this device, with this technology, you can actually address these concerns because the participant will prick their finger, and then the tip on the device will actually absorb a fixed amount of volume that's regardless of viscosity or density of the sample, and will account for variability in the hematocrit levels. Then the sample is then dried and shipped at ambient temperature, so you reduce some of the considerations that you have around traditional sampling with the storage and the shipment. Another device previously called Novaplex, and I apologize I haven't updated this image here. It's now called Telemune. But this device actually allows for automated fractionation or separation of the plasma from the whole blood sample. So the whole blood is dropped on there, and then within a few minutes, it actually separates it into the plasma, which is then air dried for ambient temperature shipping. Here are some newer wearable devices, which are also very convenient. So here on the left is the Tap 1 and Tap 2 devices from Your Biohealth. So these devices, you basically wear them on your arm, and then you push the button on demand. So there's a little green button here on the Tap device. You push it, and then it uses these micro needles to collect capillary blood collection. So it's relatively considered a lot less painful than your typical venous blood draw. And then you also have the Tasso device, which is a very popular device. Again, it's wearable. Participant wears it on their arm, and then they press the red button at the time of collection. And the device actually here depicted in the bottom center of the slide, you can see you actually collect up to four simultaneous blood samples at the same time with one collection. They also offer a Tasso SST device, which can be used to take the whole blood, and then without any coagulation can allow for immediate separation to obtain serum as opposed to whole blood. And then FluiSpotter is another wearable device. It's also a VAMS technology, but the advantage to it is that it's programmable, and so it actually allows for automated serial collection of blood samples that are then stored on this little paper roll that they use with the device. So the key there is that it allows for the serial automation of collecting blood samples. So when we think about these devices, again, there's a lot of different options already, and this field is rapidly evolving. It seems like every time I go to one of these device manufacturers' websites, they've come out with a new, 2.0 beta type device that has, improved functionality and so that it's a constantly evolving space. So, you have to think about a number of things. First of all, you want to think about for your patient population, how usable is this going to be? So, does this patient population, for instance, have caregivers that are going to help them? Are they going to collect it by themselves? Is it pediatrics where there may be concerns around the volume of the blood that you're using? Do you need from your bioanalytical perspective, do you need plasma, or can you get away with whole blood, or do you need dried or liquid samples, which again will impact, of course, how you're storing it for shipping. So you'll need to think about stability both during and after sample collection. A big one that you'll need to consider is the regulatory status. So depending on how you want to use this data, if it's going to be used for regulatory submissions, you want to make sure that it is meeting all the guidance related to that. You also want to think about how you're going to bridge. So if this is your first time doing this, you're going to have to think about how you want to bridge to your traditional, usually liquid plasma matrix for PK. And then you'll think about the volume, because most of these are very small volume devices. That's part of the advantage, but you want to make sure that you have adequate volume. And then you'll also want to think if you need replicates or multiple aliquots. So as I mentioned, the Tasso device is one where it collects multiple samples at once. I think now there's also a TAP device that also does that as well. So just some considerations that you need to be aware of as you're trying to decide what device you might want to use in your studies. So this table here, I'm not going to go through it in detail. This is taken recently from a white paper that our working group published in Clinical Translational Science. And again, this is just a snapshot in time. This is, as I said, a very highly evolving field. But these are some of the products that we wanted to summarize here, and I think it's a good reference. It gives you the names, tells you about the, whether or not it's liquid or dried, what types of matrix you're collecting, plasma, serum, blood, the volumes available, the site of collection, and some information about whether or not it's 510K cleared or CE marked for use. So definitely a useful reference here. The link is there on the bottom. So in terms of the value proposition for patient-centric sampling and drug development, there's a number of things that make this appealing, I think, to both sponsors and patients. So probably the biggest is that the concept that we could generate exposure data when it would otherwise be difficult or potentially impossible. So that could be, data for patients who live in remote locations. That can be, patient convenience, so being able to take data at home and not having to confine the patient to the clinical site. That can include, vulnerable populations where we're limited by blood volume, such as pediatrics or neonates or the elderly or critically ill patients. And then it can also include taking data at times or proximal to times of unpredictable clinical episodes. So if we think about disease states where you're thinking about the onset of an acute event, such as a migraine or an asthma attack or a seizure, or something like HIV pre-exposure prophylaxis, right? So you want to be able to potentially collect a sample proximal to some sort of event. And it can also be the onset of an important adverse event too. So it doesn't just have to be the disease state necessarily you're interested in, but you could be an adverse event of your drug-related adverse event that you want to measure exposures relative or near that event occurring. So when we think about drug development programs that are most likely developed to benefit from patient-centric sampling, one example from a PK perspective is drugs that have a long half-life. So this is something we frequently will keep people in the clinic for a long time to collect PK because we have a drug with a very long half-life, or we'll either keep them there or we'll bring them back over time throughout the study, and they'll have to keep returning to the site. This is particularly useful if you have a need to collect data at steady state, or if you're needing to confirm or evaluate that the drug is fully washed out, instead of having to bring the participant back to confirm that. You could potentially do this at home. And so this is of course important for us as a sponsor, because it increases our efficiency, but it's also really important to think about this from the participant perspective. So clinical confinement for long duration samplings is not only expensive, it's incredibly burdensome on the participants. And I cited this study here, which said that nearly 40% of healthy participants in a phase 1 trial actually self-volunteered, using an analogy of comparing their clinic confinement to being in a prison. So, that's kind of a little bit dramatic, but it is true. We keep them in the clinic for a long time, and we often control, when they can eat meals, their activity levels. Sometimes, we're collecting things like urine and other things as well from them. So in some ways it can actually, it can be very restrictive on these participants, and anything you can do to reduce that burden is significant. So it's definitely something that we should be aware of. In addition to PK drugs with a long half-life, there's also disease states where you might have a delayed pharmacodynamic biomarker that you want to collect as a response to therapy. So again, something you wouldn't necessarily want to keep bringing the participants back for, or confining them to the clinic. As I mentioned earlier, diseases where you have episodic or unpredictable clinical endpoints, such as migraine or asthma, are good examples. Diseases where you're already having difficulty enrolling, so orphan diseases, diseases where patients are, again, in geographically remote locations. This can certainly help with enrollment efficiency. And then lastly, places where there's a need to optimize clinical care or clinical trial endpoints. So this could be scenarios where you want to do therapeutic drug monitoring, and also scenarios where you might want to enhance for demonstrating patient adherence. So for example, if you had like efficacy failure in a PrEP trial, in a pre-exposure prophylaxis trial, having some sampling to demonstrate adherence could be very useful. There's also some less concrete benefits that are still, possible and are being investigated. So there's patient-centric sampling that could allow for real-world data collection, potential for collection of not just PK or PD data, but also clinical safety labs. There's simplified sample logistics. This is actually, I would say, generally already here. We have ambient temperature, shipment and storage, increased automation in the workflow. Again, ability to improve enrollment efficiency and reduce screen failures, ability to offer potentially flexible collection times. So instead of having a very strict collection time, you might need a sample at, let's say, week 24 of a study, and then you could have plus or minus a larger window, instead of having to bring someone back directly at a very specific time point. This could also lead to fewer patient dropouts and of course reduce trial costs for sponsors by reducing clinic time. But ultimately, the goal is that this would help us increase the quality and the rate of therapies that reach patients. That said, there are a number of barriers or challenges to patient-centric sampling. So one of the biggest is from a bioanalytical perspective. You do need a long lead time for demonstrating your bioanalytical assays validated and meets the requirements. You'll typically need some regulatory engagement or feedback regarding your strategy and what would be considered successful bridging for your assay. There's also logistical issues that if this is your first time doing this, you're going to have to consider how you can ensure patient compliance, storage, stability, shipment, and ensuring sample quality. You also need to consider your patient population. So is the target population or caregiver for that population, are they capable of performing patient-centric sampling? Will you need to develop any sort of specialized training for this population? So all of this really, there's a huge upfront investment in the technologies and resources needed for implementation. And so that, translates to increased cost and complexity of the protocols. And at all stages, really, there's typically a delayed or sort of undefined ROI, which can be challenging as you're sort of pitching this to stakeholders. And so it's definitely, you have to make the case, you have to advocate it for your program, and you have to consider all of these factors. Okay, and with that, I'm done, so I'll hand it off to Melanie for her side.
Great, thanks, Parag. So now we're going to talk about some of the implementation considerations. Can you click the slide? Yeah, so I mean, Parag mentioned some of these things, but as we think about bioanalytical feasibility, we have to think about the device selection, right? So think about feasibility of, can the device be used in the way that you want it to be? What's the regulatory status of it? And then planning experiments to sort of choose that device, and thinking about stability. And that stability has to be for the whole course of the sample, right? So if you're thinking remote collection, you got to think about stability there. And then obviously, if you're thinking about different populations like pediatrics, you need to make sure that you've got that sort of mapped out and considered. And then I think many of us use CROs for our bioanalytical assays. And so you have to think about capability there and whether they can support what you're trying to do, and if the skill set is properly trained there to support these. So next slide. And sampling handling, and I'll talk a little bit about this too when we go through the case studies, is also really important. So how are you collecting them? What's your training? And how are those being managed? So obviously that chain of custody is really important. And thinking through compliance and how you train to sort of mitigate those potential hurdles. Assay validation also has some things that you have to think through. You may have standards and QCs and slightly different matrices. It may be different per the device. Are there anticoagulant concerns? And then obviously, as we talk about patient-centric sampling, that's often microsampling. And so we're talking diminished volumes. And can you detect your analyte of interest? Do you have the sensitivity that you want? You also need to think about, do you have any non-specific binding? Does the device have any sort of impact on your analyte? And then automation and scalability, right? Can you really use this in large phase trials, and mapping that out is really important. Next slide. Operational feasibility is also really important, and kind of what we found on this journey is that you can, you get your assay all up and running, you think that you're ready to go, but the operational component is also a huge thing to consider. And so you have to think about the supply of it, how many devices do you need, what's the expiration of those devices, can you use the same lot if there's a volume wicking component, what's the chain of custody, labeling is important, how are you shipping to and from if you're doing home sampling, evaluation for like the global environment is really important too, because obviously the list there and the scalability is a little bit more complex. And then there's regulatory rules around like just import of different devices. And so you have to think about that when you're thinking through country selection. And obviously it's really important to think about that scaling piece, right? When you go from that small phase 1 trial to later global trials, you got to think through how to do that appropriately. Next slide. Yeah, now I'm going to shift and talk about some real world case examples. And some of these are pretty compelling. We started this working group about 2 years ago, right before COVID started. And so it was very timely, because I really do think that nothing made it more apparent that we needed as people in clinical drug development to think about doing things a little bit differently, right? And obviously the impact there on trials was huge. And so as a working group, we really started to see some really compelling case studies around applications during the pandemic. And so the first is a PK example that Pfizer has provided, and this is for the Paxlovid program, which I'm sure we've all heard about. So the phase 1 trial was conducted in March of 2021. And when they moved into their safety and efficacy trials, they needed to do those in COVID patients, but they wanted to maintain the quarantine because, patients were infected with COVID. And they wanted to, from a safety perspective, to not expose site staff and other patients to COVID. And so the approach that was taken was to build outpatient studies for their phase 2-3 studies. And the complexity there is that you don't have a very rich PK data set, right? You've done your phase 1s, but then how are they going to build that data? And so the decision, they moved to at-home sampling for support of those late phase studies. And they took a couple different approaches with home health visits. They limited site visits, and then they also looked at self-collection with the TASA device, and we'll talk through that in later slides. So next slide. And I think one thing that's really compelling about this case study is this data that was collected at home with this alternative device was actually used in registration. So I think that's a pretty compelling use case that we can use this, that it will be acceptable from a regulatory perspective. And I wanted to pause here and talk a little bit about the bridging considerations for these types of approaches. It's really important to think through your strategy on how you're going to bridge. So you've got to establish that correlation between the two measurements. So between the capillary or the venous or the plasma and the blood, you need to make sure that your data can be correlated back to what we call our traditional or gold standard values. So you got to think through when and how to bridge, and there's not one approach. It really depends on the program where you're using this. So, you may have to think about plasma versus blood, preclinical versus clinical. Are you thinking different populations? Are you going straight into pediatric? And so then you have to think about bridging different populations. And then finally, just the regular sample versus the microsampling and bridging data around that. So next slide. Yeah, and specifically in the case study that we have here, Pfizer took this approach where they did this early feasibility evaluation. And in this evaluation, they compared, venous plasma to a dried blood collection to a capillary collection of serum. And based on their results here, and I'll show in a second, they did get really good correlation between the two microsamples they used. They decided to move forward with the dried blood approach. And the reason for that is it was, minimal sample handling because it could be stored at room temperature. And it was a simpler collection for that remote piece. And so, as I said before, they looked at the 3 different sampling approaches there. This was all conducted, the feasibility work was all conducted inside the clinic. So it's a well-controlled environment to really get good, robust data on that bridging piece. They used the 2 devices highlighted here. The advantage of the dried blood was that it could be stored at room temperature. And so that really enabled that at-home collection. Next slide. Yeah, and the bridging looked quite good here between the traditional venous collection and the capillary upper arm serum, as well as the dried blood. Yeah, and the overall percent differences in bias looked quite promising as well. So next slide. And so Pfizer sort of successfully, based on this data set, decided to move forward with the TASO M20. They incorporated that into 7 subsequent trials after that. And you can see here from an assay performance, as well as from a, sample compliance perspective, results were quite promising. So overall, the passing rate looked quite good. The one study we have here that shows the lower passing rate was actually due to, some reassays that had to occur. Incurred sample analysis looked good, and overall compliance also looked quite promising. So next slide. Yeah, and we have 3 slides that show very similar things. So TASO did bridging on 3 different studies, or Pfizer did bridging on 3 different studies, and the results looked quite great in all of these studies. Biases were low, correlation looked great, and they didn't see too big of a impact of concentration on that bridging data. So here's the first case study, and then the next. And Parag, we can just go through the third. I included these so that data set would be there for people's interest if they wanted to go back and look. But overall, the results look very good. Yeah, and so in this particular case study, I think we've hit on some of those implementation considerations and challenges. But in this case study, they really lived it, and they lived it in the middle of the pandemic. And so, the device approval status is obviously really important, and that CE mark or other regulatory registration is really important. And then you have to think through, self-collection and if that's allowed in the countries that you're talking about working in. You've got to have really good partnership with your central lab. You're gonna have to rely on them a lot for supplies, kitting, sample tracking, labeling, all of that. And so building process there is important. You also have to think through what your patient-facing materials are going to look like and what translation strategy you're going to take. And site training is another piece that's very important. And so, making sure that you have videos available to people, that people get devices ahead of time at the site level so that they can really become comfortable with the devices is important. Next slide, please. And I think what this case study shows us, it's a really great and exciting one that shows that you can get reliable, high quality PK data with these approaches, and that those can be used in registration trials. The current technology and infrastructure is there for at-home PK sampling. You have to build process, but it does exist. And I do think that patient-centric sampling can become the new norm based on sort of what we've learned through the pandemic. And maybe we can have applications beyond PK into the diagnostic space, laboratory tests, biomarkers, and beyond. And I'll talk through some of those potential use cases next. Remaining challenges, they're still there. And this certainly isn't something that you want to use on every trial. But it does provide huge advantages in certain paradigms. There are some challenges around patient identification. What is the timestamp of when that sample got collected when it's collected remotely? Are there processing challenges? The hope is that integration with sort of future builds in digital health can sort of fix some of these issues. But for the here and the now, those are still things that need navigated. Next slide, please. Yeah, and so I wanted to shift and talk about a few biomarker applications that we've seen. This particular case study is out of the University of Ghent, and what they built was an assay to quantitate abnormal phospholipids formed in the presence of ethanol. So that's really about alcohol consumption, and this is really present in the urethrocytes. So you need a whole blood assay. And I think many of us know that liquid blood is a tricky matrix to work with from an assay perspective. And so they did some work looking at, could they look at dried blood, versus traditional filter paper, but then also a new device called the VEINS device, which is a little more quantitative in nature. And they showed successful bridging between the liquid blood and the dried blood. And obviously the advantage here in the dried blood is that you can then think about, sample collection occurring outside of, specialty clinical sites where you have, trained professionals collecting the blood. So next slide. And so, their next step after they showed reasonable correlation was to embark on this larger trial with 800 participants. And they did 3 finger stick collections at home. Training was all conducted remotely of these people taking that self-collection. And one thing to note here is that the enrollment in this trial took 4 days. And so, many of us talk about patient-centric sampling in terms of, well, could this help enrollment? Could it cast a wider net for our trials? And I think at least in this case study, the ability to enroll that quickly, that many participants is some indication that in certain populations, there's a lot of interest in participating in these things when we can make things less burdensome. The team actually received certification to use this method to sort of prove or demonstrate sobriety for different people. And so, the data was of a sufficient quality to get that certification. Also, comparisons in this study of the traditional blood to the work that was done in the dried blood also looked quite promising. Just another case study of a good successful use case. Next slide. Yeah, and I'm going to talk a little bit about proteomics. And this example came to us from our colleagues who's on the working group from Novartis. And they have been looking at proteomic signatures with different microsampling technologies. And the advantage here, I think, in the microsampling approaches are that you may be able to take more serial samples, right? You may be able to do more longitudinal sampling if you can think about that occurring at home. In diseased populations, that would be hugely valuable. And so that's the application and the intent here. When we think about biomarkers in general, we have to think the context of use and what sort of data quality do we need around that. And so the microsampling device pilots that Novartis did live early on in sort of just understanding what the capabilities are. And you would want to do this kind of work before you thought about using it, even as an exploratory endpoint in a trial. So next slide. Yeah, and their design was, conducted in the clinic. There was a venous collection and then peripheral blood collection using the YourBio Health device. And this is, similar to other devices. It affixes to the upper arm. You push a button, and then you get a liquid sample out of it. This device is quite painless, and there's a fair amount of data showing that. And then they also looked at how you process that sample post collection, and obviously that's really important for proteins. You don't want to be thinking you're detecting a signature in the sample, which is actually just about post collection protein degradation or changes in levels there. So next slide. The platform they used for this analysis was the SomaScan technology, which is an aptamer-based proteomic platform which measures 7,000 proteins. And so it's semi-quantitative. It's a bead-based approach. And so you get a lot of data out of these kinds of approaches. Next slide. Yeah, and what they saw is that how you process that sample really matters, right? And the centrifugation piece of when you do that can impact the data. And so it's important to sort of map out that processing piece before you would implement in a trial. Next slide. Yeah, and so when they looked at comparisons of the traditional collection to the capillary approach, there's a whole set of proteins that show good correlation, and they then took those that showed a decent correlation and mapped those and developed ELISA methods on, and we're just showing 4 of them here, which show quite good correlation. And so this is one approach where you can kind of see what proteins are going to work using different processing schemes, and then develop fully validated assays downstream. Next slide. Yeah, and then I want to shift and talk about another application from a recent publication from our colleagues at Lilly. And so what they have done is and this publication is out there as well, they did a series of experiments inside the clinic with people with abnormal liver function. And they used an upper arm device to collect a liquid serum. They did a couple different approaches on when you centrifuge that sample. And with select analytes from these chemistry panels, they showed really good correlation. And so if you're interested in that, I would dig a little deeper. There's some pretty compelling data there that shows, maybe we could be collecting safety data remotely. And the nice thing about this is, you know, we're all worried about liver tox, right? I mean, this is a big concern in clinical research. And so the ability to collect that remotely, especially in late phase trials, could be hugely advantageous. Next slide. And just wanted to highlight, you know, some further work that's been ongoing is a liver function test. And this is actually a commercially available test with Q2, and it's a partnership with Tasso. And there's 7 analytes in this, and so you can actually order this test via that central lab and get those 7 analytes remotely in the United States. So it's certainly the first step on a journey of thinking about chemistry panels and doing those remotely, but it is, you know, one of the first success cases of that. So next slide. Yeah, and so, you know, just to close it out, you know, where are the future opportunities and the benefits to these things? And I think Parag outlined this well, but, you know, really what we want to do is get more informed data, right? We're all data-driven organizations. We all need data. So can we think about getting better data out of our trials? Can we think about increasing that data quality? Can we increase access to patients by reducing burden, right? And if we think about what goes on in our trials, you know, there's people are in the clinic and then they're in the clinic again, and anything that happens in between those site visits is a bit of a black box. So if we can build capabilities to really understand the biology and the behaviors that are going on there, this could have a huge impact on research, right? Right, and the reason that we're all here, right, is to just make drug development go faster so that people can get their medicines faster. Yeah, so with that, I want to conclude and just thank everyone, Cindy and Parag, for participating in the webinar, and want to thank the IQ organization as well. This has really been an enjoyable working group, and we've had a good time sort of working through these things. And I think with that, we're happy to open it up for any questions anyone has.
Yes, thank you, Melanie, Parag, for a very informative talk. Let's move to the Q&A section, and I see already several questions lined up in the chat. So the first one is to both of you. I think this is a question perhaps a lot of our audience are curious about. What are the biggest hurdles to implementation, the greatest challenge in applying this novel approach? Maybe you can both comment from your perspective.
Yeah, I can take that one first. Yeah, so I mean, I guess because I'm a person who builds assays historically, so you know, I guess in my mind I was always like, well, if the assay works, we can get the assay working, then we're good. But the reality is it's a somewhat disruptive technology, and so there's all of this front end build. It's not really a build, but just like ironing out processes that it really has to happen. That's really about, you know, the getting the devices where they need to go, what's the regulatory status, and down to even the nitty gritty on how you label, how you package, how you store, how do you assess compliance once that sample's taken. So really it's all of these sort of operational questions that are really huge as well. And so Parag, I don't know if you want to add to that.
Yeah, I mean, I think Melanie hit the point. There's just a huge, you know, upfront investment in time, and both from the bioanalytical side, but also just, you know, the operational side. You might have to develop training specifically for this. And so, you know, at every step of the way, you are essentially, when you're trying to implement this at first, you know, you're adding some work to all the teams, right, across the functions. And you know, you're adding something that takes more thought than what we've always done. And so, you know, there's always that, I think, human nature, you know, this is what we've always done. Let's just do it this way. We have our processes down. And so, you know, I do think you get some pushback from that perspective, because the benefit is sort of more long-term, and sometimes it's more abstract too. And, you know, you have to, for instance, you know, train other functions to care about, you know, why do I care about your PK data getting better data, you know, when we do these other studies where we don't have this and it's simpler and easier to implement. And so, you know, I think that's the biggest challenge. And then, if you can find a way to really make the case, I think you have to, you have to go there and you have to show exactly what data you weren't getting that would have been, and how much that would have costed the program to try to get by, keeping people in the clinic or bringing them back. And now what data you might get, and then what would the use of that data be? So is that going to allow some sort of PK/PD modeling, for instance, that maybe will help you eventually, not have to conduct a clinical trial, or get away with demonstrating something that would have taken a separate analyses to conduct and to actually demonstrate. So I think that's sort of the biggest issue, right? Is really convincing everyone that it's worth all the effort to do this for the first time. I think once you get it down and once you've been able to do it once, then the path will become a lot easier.
Great. All right, so our next question is sort of along the same line, but a bit more specific. The question is from Mark Arnold, and it asks, how successful have you been in using dried blood spot samples in clinical laboratories? What resistance have you encountered?
Yeah, so Parag, I can let you go first on that one. I don't know if you have thoughts, but I guess I would ask a clarifying question. Do we mean clinical labs, like for safety, or do we mean just any drug development in general? I would say that, like,
yeah, in general, and just in clinical laboratories, but it's more for dried blood spot samples specifically.
Yeah. So I mean, one thing I would say is, the classical limitations are there, and I think that is, we're almost biased against it because the data is just not seen as very reliable, right? The variability that you have is well known, and so I think that's why using, if we're talking about the traditional dried blood spot where you collect it on a card, that's why there's still that bias towards this. And then if you're talking about the newer devices where, as opposed to collecting liquid blood, but we're just sending in like a BAMS collected dry blood, I think, it's an education perspective, right? You have to educate them that, these new devices are different, and they are able to actually, restrict the volume and standardize the volume of blood collected. And they can deal with changes in hematocrit and density or viscosity of the blood. And so, it's just an education, I think. I don't know that I've encountered any specific resistance to the fact that it's dry blood.
Yeah, and I would say that, like, we've definitely had a fair amount of success in the PK space doing, veins or other dried blood approaches. I think that we're starting to build the biomarker, just personally at Merck.
Yeah. Okay, our next question from Mohammed Shabli. Could you explain how patient-centric sampling could potentially increase the data quality for PK evaluation compared to traditional sampling?
Yeah, so but data quality, to me, it's data that so often when we're designing a PK study, right, we're designing sampling that is reasonable, and we're considering the logistics of keeping people in the clinic, for how long we want to keep them in the clinic just to collect PK samples. Sometimes we keep them in there several extra days, just because we need more PK data at the washout end, at the tail end of the curve. And, sometimes we send people home and then try to bring them back less frequently because, we don't want to keep bringing people back every few days. So with this, something like this, you send them the kit, it's all mailed. they could, take samples every day at home if we needed, or, every few days, and we could do that for weeks, which, if you're thinking about like a long-acting injectable drug or something like that, that data could be very useful. So that's what I think about when I think about, the quality of the data that we're able to get. That, a lot of times we're just limited by what's reasonable to do in a PK type study. PD is similar too. PD is often delayed, and you also want longitudinal data for PD often, but again, you're not always able to get that. So to me, that's what I think about in increasing the quality. The other piece is time of sample collection. So some of these devices could be paired with like a digital verification of the time that the sample is actually taken. So there's things like that could help also improve that. Maybe in a clinical site, it's not always accurately captured because it's dependent on the site staff recording that, and they're doing dozens of assessments all at once, and they're not always getting that data accurately recorded. So
Thank you, Parag. Another question from Mohammed. I think this one is for you, Melanie. So you talked a lot about the application for PK samples, also touched on other kinds of samples, such as remote safety panels. So the question is, why is the safety lab panels evaluation using patient-centric sampling lagging behind the PK evaluation? What are the major limitations?
Yeah, that's a really good question, because when I first started thinking about patient-centric sampling, like maybe 5 years ago, really in depth, I thought clinical chemistry would lead the way. It's where the greatest number of samples are. I was like, they'll go and do that, and then we and the pharma will just follow along. But I actually think that just the cost structure in clinical chemistry is so different, right? So like if you're talking about a device that's going to cost $15 on a test that they're trying to get it down to like $2, it's a no-go, right? And so I do think some of the reasons we haven't seen as much build in clinical chemistry is really just about the cost structure of that. Whereas in clinical research for us, the price of getting a phlebotomist to go to someone's house to get that sample or doing a clinical site visit is so high that we can sort of, see this as a cost savings. So I do think a lot of the investment in building some of this is coming from pharma because we need those safety panels for our trials. And so I do think it's going to end up being kind of a mix of where the investment in this is coming from. It's going to be from pharma. It's going to be central labs building this on their own because they see potential. And I do think that central labs are starting to build out these capabilities. Because, even, with the pandemic, we saw huge, increased interest in like hybrid DCTs and things like this. And it really is something when people talk about decentralized clinical trials, they never talk about the sampling piece, and it's really something that needs to co-evolve.
Thank you. The next question coming from Sally Fisher, and sounds like Sally, you've got some experience in this field. The question is, how much pushback are you getting at your organizations to implement? So maybe Melanie, Parag, you can both comment on that.
Yeah, so I think that without sponsorship, it gets really hard, right? Like these kinds of approaches, someone at a very high level has to see the value and the potential gain because, as Parag already talked, when you go to a specific trial team, even if there is this huge compelling need, you're still complicating their operational, you're adding complexity, right? And so you really have to have a good strategy. You have to have a good use case to convince, people. And then you also have to have that sponsorship piece.
Yeah, exactly. It's always easier when things come from the top. People are very good about, and I think this is just human nature, right? You're always very good about putting up walls when something becomes more complicated. And so, yeah, if you can make the case to, someone higher up, and then it comes from the top, I think that helps a lot because then barriers sort of that maybe were wishy-washy get a little, things just become easier. Some of those barriers tend to go away. And so I do encourage, also look in the literature, because as Melanie presented several examples, there's several more in the literature of other companies doing this. And so that's the other piece, right? We all, nobody, not many people enjoy being the first mover because of all the risk involved, and again, the ROI being sort of undefined. But seeing that other companies are successfully not only doing this, but using the data for, like in Melanie's example with Pfizer, right? The using this data for regulatory submission and allowing this data to contribute. I think showing that people aren't always aware of that, and so going into the literature, pulling these examples, I think it can be really beneficial to getting support.
Thank you both. Next question from Adam Bentley. What has been your experience regarding developing bridging data sets? For example, in the PK space, using a first-in-human cohort bridging to patient-centric sampling for future studies. Melanie, I think you touched on that during the presentation. Do you want to elaborate a bit more?
Yeah, so I mean, generally our strategy, well, what you want your strategy to be is you do it in early phase trials. Now, sometimes, things are coming in from the side, so you don't always get that luxury. But generally our strategy is you bridge it early in healthy volunteers, and then you move to your patient population. We've successfully done that a lot. I mean, there's been questions, I think, in the community about, like, how often do we really even see differences between populations, and is this necessary? I think there's still some unknowns about that. I mean, what we do know is that you got to bridge, you got to have a reasonable strategy for how you do that, and you've got to have defendable data, right? So, I mean, that's one thing I think that comes up is like, okay, so you have correlation, but what's the criteria for correlation? And really that criteria is like, what decision are you trying to make based on this data, and can you defend it?
So Melanie, I kind of have a follow-up question to that. It seems like in most case examples that I'm aware of, it's sort of been approached as a one-off sort of interaction with the regulatory agency around what criteria is needed to demonstrate bridging. But are you aware of any efforts, like from the agency side, in terms of creating like a guidance or anything related to microsampling bridging, or anything like that?
Yeah, so I mean, certainly something where I think there's been discussion. As far as assay regulation is included in the validation PK right now for small molecule. There's some stuff about GBS. I think for the bridging piece, I get nervous about if there were a guidance, because it's, then you're locked into that, right? And depending on how you're trying to use that data, you still be able, you may be able to defend something even if it didn't meet that criteria. So I think, yeah, that comes up a lot. And I think generally the answer I get from regulators is like, you need to be able to defend what you're trying, do you have good data to support what you're trying to do? Which isn't always that helpful.
Nice question, Parag. So that's actually take care of the next question in the queue. We are 1 minute past. I'm afraid we won't be able to get to all the questions. We still have quite a few in the queue. Due to the limit of the time, let's try to respond to the remaining questions offline. And with that, let me just thank Melanie and Parag again for a great presentation, and thank the IP organization for the opportunity, and also thank you everyone, our audience, for participating and asking the good questions. Have a great rest of the day, everyone. Goodbye.
Thank you.
Thank you.
Bye.
Goodbye.